Hi, everyone. My name is Cynthia, and I am from McGill University in Montreal. And a lot of what my lab group works on is soil ecology and soil carbon and organic matter accumulation and organic nitrogen cycling. And we really take a biological perspective when we think about these things. And the reason being is that these processes are fundamentally driven by the soil biology. How we accumulate soil organic matter, which we all know is integral for soil health, and how we mineralize that organic nitrogen is a biologically driven process. But the soil biology is really complex. And so in order to understand how we can build our soil organic matter and increase nitrogen mineralization from those organic nitrogen sources, we need to understand something about how the soil biology is functioning in our soils. And one of the reasons, I'll just operate two of these at once. Okay. The soil biological community is so complex is that it's incredibly diverse. It is the most diverse ecosystem on the planet. And that's because we have all of these different organisms. We have bacteria. These are bacteria colonizing fungal hyphae. We have protozoa. These graze on bacteria. They can even graze on root tips, for example. And then we have our soil fauna, which help break down your litter, and also our sort of soil engineers in moving soil particles and litter through your soil from the surface down into soil depths. And so all of these organisms are key players in a lot of important ecosystem functions that we care about. But there's an incredible diversity in these systems. So in a single teaspoon of soil, I'm sure you guys have heard all of this before, but there's millions of individual organisms, millions. So this is a very diverse ecosystem. And what these organisms are doing is they're carrying out some of our most important functions that we think of when we think of a healthy soil. So one of my favorite topics is soil carbon. Which is most organic matter. 60% of organic matter is carbon, and it's the major food source for your microbial community, and so it's a very important component of your soil. And the soil microbial community is both breaking down that carbon, but it's also building your carbon. And so they're a very important component of soil carbon cycling. And then, of course, the suite of diversity, of metabolic diversity of the microorganisms can help remove toxins from your soil and help remediate soil and help remove heavy metals from the soil. And we inoculate soils that have had oil spills to help clean up oil spills. And so these organisms can be very good at degrading compounds that we don't necessarily want in our soils, and they can even degrade some pesticides. And so they can be very good at removing any pollutants or toxins that we don't want in our soil that can accumulate, bioaccumulate in plants, which we don't want, right? So this is a very important service. And then, of course, all of these bacteria can help provide disease suppression. And so the more diversity we have, the more likely we can achieve a more disease-suppressive soil. And then, of course, it's these organisms that are driving nutrient cycling. So if you're not adding mineral sources of nitrogen or phosphorus, all of your other nutrients are then coming from organic sources, and it's the microbial community that's making it available for plants to take up. And so they're very important for driving nutrient cycling. And what's really cool about a lot of these microorganisms is they can actually communicate with plants to help stimulate plant growth promoting hormones, for example, that can allow plants to overcome a lot of stress factors like drought and heat stress. And that's between the communication of plant roots and the microbes. And so there's some really cool things that this diverse system can be doing in your soil. And so in terms of soil health, what we often think of is the more diverse your system, the more of these different organisms you have, the healthier your soil is going to be. So a healthy soil is a diverse soil. And there's a lot of evidence to support that, and I would suggest that that's often the case. And this is, for example, what I'm talking about, whereas we increase on the x-axis soil biodiversity. And so in this case, biodiversity means the individual different types of taxa. So if you don't like to think about bacteria, you could think about this in terms of crops. So increasing crop diversity would be increasing the number of different crop species you have in your rotation, for example. And so this is the same thing, but in this case for bacteria. So the number of different species of bacteria and fungi we might have in the soil. As we increase that, we're likely to see an increase in soil function, say, for example, nitrogen mineralization. And part of the reason is that the more species you have in your soil, the greater the odds that there's going to be a species there that can carry out the function that you care about. And so you're just increasing the odds that the species that are responsible for a lot of these soil services are present. And then also, if you disturb your system, or there's some sort of or there's a dry spell, or you till your soil, for example, the more diverse your soil is, the more quickly it can recover, and the more likely, if you lose a certain species, you're going to have another species there that can still carry out that function, because there's some overlap in the ability of bacteria and fungi to carry out the same function. And that's very good. The more overlap in function there is, the more resilient your microbial community is going to be. And so this relationship, we often think about this relationship in terms of soil health, but there's something that we overlook, and that's that a lot of the organisms, as we increase in diversity, are not actually active. And so when we think about our soil biological community, especially the microbes, most of them at any given time are not doing anything. They're what we would call dormant or metabolically inactive. They're sleeping, and that's because the soil is a really harsh environment for the microbial community. Whether you're in a forest system, or you're practicing regenerative agriculture, or it's an intensified monoculture, it's a very harsh environment. And so most soils, for example, are really carbon limited, and carbon is the most important energy source. It is the energy source for most microbes in our soils, but most of these microbes don't have a lot of access to carbon. And so that's one of the constraints. And then sometimes there's constraints with flooding. So microbial activity is often depressed under flooding conditions. It can be depressed under changes in pH, and we can get microscale changes in pH just as we move away from the root zone. We can get very acidic conditions at scales that microbes are experiencing. And so in a lot of cases, our microbial community, while it might be diverse, they might not be able to carry out those functions because they have some sort of constraint, because these are not very hospitable environments for the microbial community. And so one of the approaches I like to think about is, while this is an important relationship, is to actually think about how we can improve the existing community, how we can improve the performance or capacity of the microbial community that's already there. And so we can think about this same relationship, but instead of soil biological diversity, we can think about this in terms of soil biological performance or fitness. So as we improve the overall capacity or fitness of the microbial community that's already there, we should expect to see an increase in our soil function, because now the microbial community is more active, it's growing, it's doing the things that it's supposed to be doing. And so one way to think about this, if you, again, don't like thinking about microbes because you can't even see them, is think about a sports team. So if you're a coach and you inherit a new sports team and you have this group and they're pretty lazy, and you go through the first season and your team does terrible, right? So they lose all their tournaments. You have two options. You can fire everybody and get a new team, and that's one approach, and that would be something like changing your community composition in your soil. Or you can train your team. You can bring in experts in certain I don't know very much about basketball, but like you can bring in a coach just for your point guard, for example, and you can really drill down on expert training. You can give your team the resources they need. You can buy fancy equipment, and then maybe next year, their performance is better. And so that's what we're trying to do here, is rather than changing our team, we're just trying to improve the performance of our team. And to me, this is a much more tractable, manageable approach for improving sort of our healthy microbiome for soil health, is really capitalizing on who's already there and giving them the tools they need to really improve their performance. And in the case of bacteria, this is really the main message that I want to get across today, is it's carbon. And so what microbes are really lacking in soil is carbon. And so we can imagine that if we want to improve the performance of our microbial community, we need to give them their energy source, which is carbon. Now, that's partly going to depend on different types of carbon. And so we'll talk about that a little bit. But if we give the microbial community more carbon, we might be able to activate some of those sleeping microbes and see an increase in function and performance. So, given this, I want to hopefully talk about 2 case studies where, rather than looking at, say, a change in microbial community diversity, we've changed the performance or fitness of the microbial community that's already there. And so in the first case study, we're going to look at nitrogen cycling from organic sources and how that's influenced by root biomass allocation. And then in the second case, we're going to talk about how we can accumulate soil carbon, not by modifying our microbial community composition or diversity, but improving their growth under these cover crop organic systems. So we'll talk first about this case study. But to review, since we're talking about the mineralization of organic nitrogen, I want to spend a brief moment talking about what I consider the biological nitrogen cycle, or the nitrogen cycle that's really driven by the soil biology. So how many of you add any organic or rely on any organic inputs for your nitrogen or fertilization? A little less than half. So how many of you think that if you're not adding an organic nitrogen source, that your plant nitrogen, some of that plant nitrogen is coming from an organic nitrogen source? Does that make sense? Yeah. So this, and this is often the case. So a lot of organic farmers, they're of course strictly relying on this organic nitrogen pool. But even in heavily fertilized systems, a lot of your plant nitrogen is coming from organic nitrogen, from your soil organic matter pool. And we see sometimes in the Midwest that 50% of corn nitrogen is coming from the mineralization of your soil organic matter pool, and that's a lot. And so this is an important pool of nitrogen for all of our growers. And so the challenge is, how do we get this organic nitrogen into a mineral form that the plants can take up? And like I said, this is a biologically driven process, and so we need to understand what the limitations are here for the biology. And so it starts, we can add organic nitrogen inputs. This could be crop residues, compost. Roots are an organic nitrogen input. They add amino acids to the soil. We have our background soil organic matter. That's an organic nitrogen input. And then what happens is that organic nitrogen is used by what we consider soil decomposers. And this is really a general class of organisms in the soil, and that's because a lot of organisms are capable of decomposing organic nitrogen and organic carbon. And so it's largely done by our fungi and bacteria, but they take up that organic matter as their primary carbon source. That's their energy source, and they're using that nitrogen as their nutrients. And then, of course, what happens to that is when this community that's taken up the organic nitrogen, it's converting it into ammonium within their cells. And so when they die and turn over, they're releasing ammonium into the soil. And so this is where we get this accumulation of ammonium, and this is where we start to get the mineralization of your organic nitrogen. So first it has to pass through the decomposer community, and then when the decomposer community dies and turns over, it's releasing that internal nitrogen back into the soil environment, but as ammonium, something that the plants can now use. And so we see some uptake of ammonium by plants. And then another biological process is, of course, bacteria can convert this ammonium to nitrate, and this happens very quickly in agricultural systems where we get ammonium going through a couple steps and then ultimately to nitrate that then the plants can take up. And so you can see this is sort of a loop here, but what I really want to focus on here is this process, because this is really the rate limiting process. These are the organisms that are going to determine how much ammonium ends up in your soil. And this process is really driven by enzymes, which I'll describe, and it's also really limited by carbon, because these microbes need carbon to grow, and if they don't have carbon to grow, then they're not breaking down this organic nitrogen and converting it into ammonium. And so if they're not doing anything, then this step isn't happening. And so we want to increase the activity of these guys. And so part of what they're doing is they take these large polymers like leaf litter or something, and they can't bring that into their cell wall. They need to break it down into something that can pass through a cell wall. So what they do is they exude these enzymes, and like our own bodies, we have enzymes in our gut. So when we eat high fiber cereal, we need help digesting it, and so the enzymes in our guts help digest that so we can access the energy and nutrients from our cereal. So microbes are doing the same thing, except their enzymes are on the outside of their bodies, and so they're basically digesting their food in the soil environment by producing these enzymes. And so they'll exude these enzymes, and the enzymes will attack leaf litter or roots or other organic nitrogen in the soil, break it apart into small soluble compounds that are dissolved and very much smaller, so the microbes can then pass it through their cell wall. And that's the carbon and nitrogen that they're taking up. And so there's a step here where the microbes have to release enzymes in order to access this organic nitrogen. And this is important because if the nitrogen and carbon is already in this form, if it's already in this soluble form, then they don't have to go through that long process of breakdown with enzymes. And one of those sources is from roots. And this is where roots become very important. So this is our soil rhizosphere, and this is just an example of maybe what a soil rhizosphere looks like, and there's various definitions, but we tend to think of it as any soil that's kind of attached or stuck to roots, and this is a super dynamic zone. There's a lot of chemical changes going on. Like I mentioned, you can get changes in pH and oxygen levels just right within this zone, which is often less than 2 millimeters, and this is also a zone of elevated microbial activity. So we have high rates of nitrogen cycling right in this zone, and overall fewer organisms are dormant and fewer of them are inactive right in this zone. And the reason is that these roots, one of the reasons is these roots exude and release what we think of as root exudates, and these are soluble compounds. So the microbes don't have to break down these compounds. They're soluble organic acids, they're sugars. Microbes do really well with glucose and sugars, and amino acids. And so these are compounds that the microbes can take up almost immediately right into their cell. And so they don't have to waste energy exuding enzymes, and it's also a very rapid process. And so what happens is the roots exude these small compounds, and then it stimulates. Here our microbiota, it stimulates the microbiota because now they're no longer carbon and nitrogen limited. And then what happens is when their growth is stimulated, then they can produce more enzymes, and this can start attacking your larger organic matter, so your compost and crop residues. And it can almost prime the breakdown of more complex, lower quality crop residues, for example, or other types of organic inputs, because you have this actively growing community that's no longer carbon and nitrogen limited when it's getting these soluble compounds from the roots. And then, of course, because of this, we see an increase in plant available nitrogen that the plant can then take up. So what we have then is sort of this pump, the rhizosphere acts as a carbon pump. So we've got a lot of carbon going into the soil where the microbial community is living around the plant roots, so alleviating that carbon constraint that a lot of microbes face in soil. And consequently, it stimulates organic nitrogen mineralization because it's stimulating the microbial community that's responsible for this. And then we get an increase in the release of this plant available nitrogen. So this is an ideal scenario, but part of the problem is a lot of our plants don't have extensive root systems, and we don't always have living plants in the ground. And so one of the emerging tenets in soil health is to keep a living plant in the ground, and this is one of the main reasons why. That living plant is going to help maintain the activity of your microbial community. that's a challenge, right? It's a challenge to bring in perennials into your rotation. It's sometimes a challenge to bring in cover crops. And so we have other opportunities by maybe selecting for different crop varieties that are actually have differences in the root biomass, and which could expand this zone in your soil. So rather than say increasing your rhizosphere and living roots temporally, you can increase your rhizosphere and living roots spatially in your soil. And so to get this, you need lots of living roots, but you also need an abundance of microbes already in your soil that can be turned on or sort of activated. And so you also need to think about what sort of your background bulk soil microbial community is doing. If there's not a lot of organisms there to turn on, then this effect of the rhizosphere is going to be lower. So this gets into one of these experiments that we did where we looked at changing root biomass based on genotypes and different wheat cultivars. And so this is just a cartoon of what we were thinking. Is this is an idealized situation. This is corn, but we used spring wheat. In an idealized situation, like I said, you've got a big root system where you've got a lot of allocation going below ground, and then consequently you have this carbon pump and stimulating organic nitrogen mineralization. We could have another scenario where we have a different corn variety with a much smaller root system. So root biomass allocation is much lower, and so the volume of this active rhizosphere is smaller, and you're not sort of taking advantage of a larger portion of your soil that you could potentially activate. And so your carbon pump is lower. You also don't have sort of an abundant microbial community in the bulk soil to turn on. And then consequently you have lower ammonium and nitrate mineralization. So in another scenario, you might have something where you do have an abundant sort of diverse and large microbial community. So if you're adding a lot of compost to your soil, this might be the situation where if you take a soil sample and you look at its diversity and microbial biomass, it's quite high. But if you have a root system that's very small that you are not using to really activate and turn on this abundant community down below the root system, you're not taking full capacity of the organisms that are down below or outside your root zone. And so this is something we wanted to explore, really to determine if the effect of wheat cultivars or varieties with greater below ground carbon allocation had a greater nitrogen cycling response to compost treated soils. And so we did this in a greenhouse. This work was led by June, who, well, now he works in oil palms in Indonesia, but he was working in spring wheat in this case. And we had 4 varieties of wheat and a wild accession of wheat. And so we took these wheat varieties that had this gradient in root biomass, and then some were we had each set, and one set had compost added and one set did not. So we had a treatment with compost and a treatment without compost, and this was the main nitrogen source. And so over time, we measured this rhizosphere to see what was happening with nitrogen cycling. We looked at those enzymes that are involved in the breakdown of that organic nitrogen into those soluble compounds, the rate-limiting step in organic nitrogen mineralization. And then we looked at microbial biomass carbon and nitrogen. And so the higher your microbial biomass nitrogen is, the more you're going to have released into the environment when that microbial biomass turns over or dies and releases it. So the higher this value is, the higher your microbial biomass nitrogen is, the more likely you're going to see high levels of soil ammonium. And then we looked at community diversity through some genetic sequencing, and then we also measured some plant variables, particularly yield and various root traits and root biomass and above ground biomass. And so just to convince you, this is a small number of cultivars we looked at, but across the 5 that we used, there was this clear gradient in how much photosynthetic carbon was being allocated below ground. So we have, across these 5 genotypes, we have differences in total root biomass on an absolute basis, and then we also have a difference in the relative amount of root biomass relative to, say, above ground biomass. So this red 5 here, this would have much more above ground biomass relative to below ground root biomass. And so we have this clear gradient, and so the next question, is to see what the effect is going to be on nitrogen cycling. The first thing we want to look at, though, is are these changes in root carbon allocation impacting microbial community diversity? And so this is something we think about a lot with soil health, and what we find is that there's no effect of root carbon allocation. As we increase root carbon allocation, we don't see any change in diversity. And so this is just the number of different species that we find in the soil, the number of different bacterial species we find. And we don't see any change. And we don't even really see a change between the compost and the no compost soil. So there seems to be no treatment effect on microbial community diversity, either by genotype and root carbon allocation or by compost additions. And we can look at some of the bacteria that are really responsible for driving the nitrogen cycle. So these are 3 different types of bacteria, and we also don't see an effect of the relative abundance of these different bacteria in terms of root carbon allocation. And so the point here is that we have these different varieties with different root carbon allocation, and it's not changing community composition, it's not changing community diversity. But that doesn't mean it's not having a positive or negative impact on your system. So we can look instead of at who's there and how many species are there, we can think instead, what are they actually doing? And to me, this is far more valuable if we're interested in soil function to actually go out and measure those soil functions. And so what we find is these are our enzymes. So these are the enzymes that the microbes exude to break down that organic nitrogen, that first step in nitrogen cycling. And this is a carbon cycling enzyme. Again, we don't see any impact of root carbon allocation on the activity and production of these enzymes. So regardless of the type of variety we use, we don't see any change in these enzymes. But when we, that was without compost. So in the soils that were not amended with any organic nitrogen. When we add compost, though, we start to get this effect of genotype emerging, whereas we increase root carbon allocation, we start to see an increase here. These are the genotypes with higher root biomass. We see an increase in enzyme activity. And so what this is saying to me is that something is happening with these varieties that have more root carbon allocation, that when there's an organic nitrogen source provided, that they're stimulating their enzyme activity. And just keep in mind, these enzymes are the first step in mineralization of organic nitrogen. So without these enzymes, you're not going to get organic nitrogen mineralization. And so we're getting an increase or stimulation in these enzymes as we increase root carbon allocation. And so if you think about, go back and think about those root exudates and those soluble compounds that roots are releasing, those compounds could be helping to stimulate the microbial community and allowing them to increase the release of these enzymes. But it's only going to happen when you have compost, because obviously you need a source of organic nitrogen for the microbes to break down. So without the compost, we just have mineral nitrogen, and there's no reason for the microbes to be active because there's no organic nitrogen. So they might take up some of the inorganic nitrogen, but they're not going to be actively breaking down organic nitrogen when it's not there. So if we look for also at microbial biomass nitrogen and our soil ammonium pool, we also see a similar trend where when we add compost, we start to get this effect of root carbon allocation on increasing microbial biomass nitrogen and on our soil ammonium pool. The effect is a little bit smaller, but we start to see an emergence of how these root carbon allocation is affecting the accumulation of soil ammonium. And then importantly, we also looked at wheat yield, also protein content, and a number of other things. And to me, this is what's really interesting is, again, we see a slight decline in yields as we increase root carbon allocation. This might make sense to you if you're increasing root carbon allocation. Maybe you're sacrificing above ground biomass and seed production. And so you might expect a slight decline. And we do see a slight decline when no compost is added, both in wheat yield and in really no effect in above ground biomass. But when we add that compost, now we see that the genotypes with more root carbon allocation are now outcompeting the genotypes with lower root carbon allocation when we add this compost in terms of yield and biomass production. And so in this scenario here for the red fife, we have low root biomass allocation and we've added carbon, but it's not going to matter so much because there's not a lot of roots there to break down this compost, to help facilitate breaking down this compost. And so when we have both roots present and compost is where we start to see this stimulatory effect of roots. And so this is sort of a summary of that data I've showed you, where as we increase root carbon allocation, when we don't have an organic nitrogen source, root carbon allocation doesn't really seem to matter as much or at all. But when we increase root carbon allocation with compost, we start to see this interaction between the soil, the roots, and the soil microbial community, where we get this stimulation of nitrogen cycling. And so to summarize, this experiment that we worked on is first that we do get differences in root biomass allocation across varieties. So if you don't have options for changing your crop rotation, for example, you can increase your root biomass by potentially considering other types of varieties where there's greater root carbon allocation just due to differences in breeding and the varieties you choose. And that when we do see these differences in root carbon allocation, we're not necessarily affecting diversity. We could affect diversity, and there are some studies that show this change in microbial community diversity, but more importantly, even if we did see a change in diversity, it's not going to tell you that you're affecting end cycling. And so what we do see is that we're shifting the function in these systems when we have a change in root carbon allocation. And that the genotypes that respond the most to compost additions are those with higher root carbon allocation. So when you're adding compost and you are hoping to see a high response, and especially in terms of nitrogen mineralization, that seems to be occurring more so with the genotypes that have more root carbon allocation. Yes, sir. Yes, so that's a great question. So how well would this relate to, so instead of using organic nitrogen, synthetic fertilizer? So my guess would be you would see something like this, because with the mineral fertilizers, there's no incentive for the microbial community to break down organic nitrogen, or there's less incentive. And so there's two things happening. One is that those roots are not as important, because when you have a large pool of ammonium already, those root exudates, they are important, don't get me wrong, they are important for a lot of reasons, but in terms of this study, those root exudates are not going to be as important in terms of stimulating the microbial community, because the plants already have access to ammonium nitrate. Does that make sense? Right, and that's a good point. And so we do see a lot of organic nitrogen being taken up under mineral fertilized conditions, and so you do want to stimulate that pool, but unfortunately, typically what we see when we, under highly fertilized systems, we tend to depress microbial biomass. And people think that has something to do with the salts that accumulate, but and the processes that the microbes are doing, changing the pH. But so it's a balance between a small amount of fertilizer or integrated mineral fertilizer with your organic nitrogen pool. But under intensively fertilized, mineral fertilized systems, this function is going to be harder to achieve. It's not that it's not happening, but my guess is that that interaction between the roots and the microbial community, there's less of a necessity for it from the plant perspective. Absolutely, yeah. So we use compost in this study, but it could have been crop residues, it could have been corn stover, it could have been root residues, dead roots, it could have been anything. Okay, so the second case study I wanted to talk about is soil carbon accumulation and soil organic matter accumulation. And because we're at a soil health workshop conference, I don't think I need to go into the details of why soil organic matter is important for soils. I think we all know that soil organic matter is fundamental for water holding capacity and for improving soil structure and soil aggregation. It is the food source for microbial communities. It provides nutrients for plants. So there's a whole host of benefits for soil organic matter that I hope all of you already are convinced of why you want to build your soil organic matter pool for a healthy soil. But in order to build soil organic matter, we have to have some understanding of how it's formed and how do we build it. In order to build it, we have to know somewhat how it's formed in the soil. And so I've simplified this into what I consider two very basic pathways. And the consequence of these two pathways is two very different types of soil organic matter. And this soil organic matter is going to provide different properties or functions to your soil. This is probably what most of you are familiar with. So we add organic inputs to our soil. We add leaf litter, we add crop residues, cover crops, compost. And this is the primary way we're getting organic matter back into our soil. And what happens is this decomposes and it ends up in our I said non-living soil organic matter pool because this is dead, but we also have living biota that make up some of the soil organic matter pool. But this stuff gets partly decomposed, and what doesn't get decomposed accumulates as soil organic matter. So that's the basic principle of how we might build our soil organic matter. You add inputs, some of it decomposes, some of it doesn't, and what doesn't accumulates as your soil organic matter pool. And this is a very important process, but the microbial community, as we know, exudes these enzymes and they break down this soil organic matter. And indeed, what we're learning about soil organic matter is that microbes are incredible decomposers. They can break down almost anything you throw at them. I mean, how quickly does your soybean residue disappear from your soil surface? Pretty quickly, yes? And even corn stover and things that we think microbes can't degrade very well, it's being broken down, but it's really contributing to the short-term pool of your soil organic matter. It's an important pool because it's the pool where a lot of nutrients are released and it facilitates aggregation and things like that. But because microbes are so good at breaking this stuff down, it disappears very quickly. And so it's really not contributing to some of our most stable, long-lived soil organic matter because microbes are eventually breaking down all of that plant material, especially our crop residues and plant inputs. And so there is this second pathway as these microbes break down and decompose your crop residues, they're using that carbon and that nitrogen in that plant material to build their own biomass. And there's carbon and nitrogen in their own biomass. And there's trillions and trillions of microbial cells in the soil. And what happens is they don't live forever. They die, just like plants and our dead plant material that we add to the soil. As this living microbial biomass pool dies, all of those cells are now entering your soil organic matter pool. And it is a significant portion that ends up going into this non-living soil organic matter pool. So what we have is the microbial biomass taking up this plant litter, converting it into their own carbon-based biomass, and when they die, they're ending up in this non-living soil organic matter pool. And what's different about this bottom pathway is that this tends to stick around much longer, and that's because microbes are so good at decomposing almost anything we throw at them, especially in these temperate, disturbed systems, that the carbon and organic matter needs to be protected. So whether it's in an aggregate, but more importantly, a lot of this carbon gets protected on clay minerals and soil minerals. And so what we find, this is an image of, this is a microbial cell. We find these cells sticking really, really tightly to clay minerals. And because of that tight adhesion to clay minerals, this microbial biomass is accumulating over time, and it's not likely to be lost like a lot of this plant input. And so the key here is that to really create this persisting, long-living soil organic matter pool is to shunt or move a lot of this plant material into the microbial biomass so that it can go through this pathway and accumulate on clay minerals and get protected inside aggregates. So this pool, this microbial residue pool that sticks on clay minerals, we now think is what's really accumulating as our long-term persistent soil organic matter. And it's part of what's, and surprisingly, it can actually accumulate quite quickly because the microbial cells turn over very quickly, and so it's just a matter of time for those microbial cells to build up to something that we can detect on an instrument. So the question here is, we know this pathway exists, so what is happening with the living microbial community? So we have all of this microbial biomass that's in the soil, maybe half of it's asleep, maybe half of it's kind of somewhat active. The more we can increase this living microbial biomass pool, the more we can increase this pathway. So in the same way we think about the more above ground biomass we have planted, the more we can add back to the soil, or the more residues we leave on the soil surface, the more input is going back into the soil organic matter pool. The same is true here. The more you build up this pool, the more you're increasing this input. And so one of the important drivers of this are how microbes are growing in terms of their efficiency and their growth rates. And so just like you and I, we all grow differently and we use our food differently, and where those calories go differs between people. Microbes are the same, so they have differences in their growth rate and their growth efficiency, which fundamentally affects how quickly they can grow. And so pretty much we can manipulate this so that we can increase this pool. And the short story here is the easiest way to manipulate this and increase this is to give the microbes low carbon and nitrogen inputs and give it to them frequently. So frequent carbon inputs that tend to have a low C to N ratio is really going to stimulate how microbes are growing, and it's going to increase this microbial biomass pool, and then eventually increase your soil organic matter. So we can change the capacity of these microbial communities in terms of how they're growing and really facilitate this pathway. Okay, so very quickly, what specifically do I mean by this? This is microbial carbon use or growth efficiency and microbial growth rate. This is a cell. These are two different cells. Each bacterial cell is taking up a soluble carbon compound, and a bacteria with a low growth efficiency means more of that carbon that it takes up through its cell is going to be respired. So just like all of us, we respire CO2 when we breathe. Microbes are doing the same thing. They're respiring carbon dioxide. So for every unit of carbon that a bacterial cell takes up, more is lost to CO2 instead of accumulating in their biomass, and that's a low carbon use efficiency or growth rate. And so you can imagine that the higher this is, the more biomass you're building. So for a high growth or carbon use efficiency, the more carbon is being used to build biomass, and the less is being lost as carbon dioxide. And then we have microbial growth rate, and this is simply the time that it takes for a microbial community to grow. So for any given time, a high growth rate, we have more bacteria being produced over any given time. So the higher the growth rate, the more numbers of bacterial cells you're producing. And so to summarize this, a higher carbon use efficiency means that more carbon that a microbe takes up is being used to build new biomass. And if we want to build new biomass to build our soil organic matter pool, this is something we want to target. And then for a higher microbial growth rate, this just means more new microbial biomass is being produced for any given unit of time. And what we know is this varies a lot across fields, but generally as we increase our input quality, so we increase access to legumes and low C to N inputs, and we increase the frequency of those carbon inputs, whether it's through root exudations, having living roots in the ground, small input rates, small but frequent input rates of compost and manure applications, we tend to see these higher growth rates and growth efficiencies in the soil. And so you can think about this, say, in terms of a hairy vetch and wheat. And if you plant these two different crops, you might select for very different communities with very different functional capacities. So in this case, we have one type of bacterial community growing under vetch, and here we have a different bacterial community growing under wheat. And they're going to differ in terms of how much biomass they can grow. So wheat might facilitate more or a higher carbon use efficiency or growth rate because it does have this lower C to N ratio that's going to improve growth efficiencies and growth rates. And so consequently, you might expect that you're going to get more of these microbial cells turning over and accumulating in your stable soil organic matter pool. And so we can really facilitate this pathway by changing the resources that microbes have access to. And so we tested this in a field in Michigan, and this is a field that has had some unique things going on where it's been in conventional and organic agriculture since the 80s. And this is showing for the conventional site the soil organic carbon. So the conventional site over time has been losing carbon. This is showing the change over time since 1989. So this is annual net primary productivity here and soil carbon here. We set for the, this is switched, but this is the conventional system. It seems to be losing carbon over time, certainly not accumulating carbon, but the organic system is slowly accumulating carbon over time. So we have slightly higher carbon soil organic carbon in the organic system compared to the conventional system, but we have lower carbon inputs in the organic system. So what is one of the defining features of organic systems? Well, that is partly why we see total lower carbon inputs is lower production, but the other component is we tend to add a lot of organic matter, external organic matter, whether it's compost or manure. We add a lot of organic matter to organic systems. So generally speaking, organic systems tend to have higher soil organic matter, but due to higher total carbon inputs from external sources. But here in this system, no manure or compost is added. So these systems are not getting any external fertilizer. So all these carbon inputs are coming strictly from plants, both below and above ground. And so what we find is because it is slightly less productive is we have slightly lower carbon input. So we have fewer carbon inputs going into the system, but we still have carbon accumulating. And so what's going on here? So these systems are in a corn, soybean, wheat rotation, and the organic system does have this rye and clover cover crop. And so we included the carbon inputs from these two cover crops in this figure here, and we're still not seeing higher carbon inputs with the cover crops. The other main difference between these systems is the organic system gets much more tillage for weed management. And so the aggregation between these two systems was not really that different in terms of the number of aggregates, and the organic system's getting more tillage. So here we have a system where we would think it's not accumulating carbon. We have lower carbon inputs, higher tillage, no change in aggregates, but yet we're accumulating carbon over time. And so, thank you. Our thinking is that what we're doing is we're increasing input diversity and we're increasing the frequency and the quality of inputs. So by including these cover crops, we're lowering the total quality of inputs by including this red clover. in the system, and we're also now increasing the frequency, or the frequency that carbon is added to the microbial community. So during these periods where the cover crop wouldn't be planted, in this case it would be the fall and spring, the microbial community is really shutting down because they have no carbon to access when the fields are fallow. But here, with the cover crop, now we've given the microbial community a carbon source, and they might be able to maintain that elevated growth rate and growth efficiency when these cover crops are planted. And so the thinking is that just by adding these cover crops, that we're actually, rather than altering microbial community composition, is that we're able to overcome these reduced carbon inputs by changing how the microbial community is growing, by improving their growth efficiency and their growth rates. And so just very briefly, we went out and measured these things, and we do see for the organic system, we have higher carbon use efficiency throughout the 3 time points we measured. And so remember, the higher the carbon use efficiency, the more microbial biomass is being built by the more carbon that's being used is being converted into microbial biomass. And then similarly, if we look at microbial growth rate, we also see higher microbial growth rate during the peak growing season, less so in September. So it appears that these organic systems, the community is growing more efficiently and it's growing faster in these fields. So then what we did is we looked at how new carbon that we're adding to the system is the transformation or fate of that carbon that we're adding. So in this case, we used something we could easily track, but you can imagine this is something like a crop residues or roots, for example. And so what we did is we added new carbon to the soil, and we could follow its fate because it had a label. And we find that more carbon in the organic system is taken up, more new carbon in the organic system, so carbon we added that spring, is taken up in the microbial biomass, which we'd expect because we have that higher carbon use efficiency and growth rate. But importantly, even though there's more uptake of that carbon, which you might expect would facilitate faster decomposition rates, we're still seeing more of that new carbon in the organic system, especially in these fractions where we know the carbon tends to accumulate and be protected the most. So we think about our aggregate fractions and our clay fractions, where that microbial biomass really sticks. And so we're getting more carbon uptake by the microbial community, and after 12 months of adding that carbon, we still see more of it persisting in the soil. And so what we think is happening here is that, well, first of all, we see this really nice positive trend between carbon use efficiency and percent soil organic carbon. So as we're increasing carbon use efficiency, we're increasing soil carbon. And so to summarize all of this, it seems like with these organic cover crop systems, we can't be sure if it's the cover crops per se, but to me it seems like the most parsimonious explanation is that we're increasing carbon growth rates and carbon use efficiency, how the microbial community is allocating whatever carbon input you're adding. So it's changing what that microbe is doing with that carbon. And it's facilitating more microbial biomass that can then end up in the stable soil organic matter pool. You have high pH? Yeah, so similar to plants, most microbes are going to do best at neutral pH. At really high pH soils, you're going to start losing some of your fungal population and start favoring a more bacterial dominated system at a higher pH. But if you really want to build up that overall microbial biomass, a neutral pH is best.